and take a few seconds to sink in. Let your mind get ready. Get comfortable. Whatever it is that you need to do to prepare. Welcome back to Physiology. Talking about cell-to-cell communication today, focusing on how do cells talk to each other, because that's kind of a big part of everything. How our body works is how cells talk to each other. We're going to talk about structures first. There are some specialized type of cells that have proteins between them. That would be these guys. These blue structures. They're like tubes between the cells, and we're going to be talking later when we get into different systems about where exactly we find these tubes between cells. These tubes, many of them, form a structure between cells that are called gap junctions. And these gap junctions allow small particles to just flow freely from cell to cell, and this is critical to the function of many of our body systems. Next is cell-to-cell binding. What we have is cells actually having to physically come in contact with each other. We have this part of this cell has to physically come into contact with this part of this cell, and this is really prominent over here in the immune system. So getting the immune system up and running. When I was talking to you before, when we talked about getting B cells activated, what this is showing is that actual process. This cell-to-cell binding is critical to the function of your immune system. Extracellular messengers, this is when a cell releases something that's going to go and interact with another cell. Sometimes that's something close and sometimes that's something far away. Hormones are extracellular messengers, and we're going to spend several lectures talking about the endocrine system, of which the focus is hormones. The little microscope here is just indicating that this person's cells, or this cell is not actually this large in comparison to this shadow person. Hormones are special in that when the extracellular messenger is released from an endocrine cell, so for example here, it travels a distance. So hormones are released into the bloodstream and they have to travel a distance far away from the endocrine cell that released them. And then they have some sort of action on a specific receptor, a receptor that's specific to that chemical messenger that got released into the blood somewhere far away. And once again, I've used a little microscope to indicate that we're zooming in on this cell. So hormones release an extracellular messenger. They release it into the bloodstream. It has to travel far away and interact with its specific receptor. We're going to come back to that. Neurotransmitters. So this is a neuron. We've seen this before. And what happens is that this neuron is going to release an extracellular messenger here at this end, and we'll talk more about all these different structures. This might be familiar, but it's going to release an extracellular messenger, and it will release an extracellular messenger in close proximity to its target cell. So neurotransmitters are both released by neurons and released close to their target cell. That's different from hormones. Or hormones has to travel a far distance from the endocrine cell that releases it. So it's released, hormones are released by an endocrine cell, has to travel a far distance in the bloodstream, whereas neurotransmitters are released by neuron, released close to their target cell. They don't have to travel in the bloodstream. Okay, so different extracellular messengers, as we just saw when we're discussing hormones versus neurotransmitters, can have actions in different places. Local mediators are ones that are released close to their target cell. A local mediator, specifically that has an action on a different cell. So here, what these little triangles are indicating is they're flowing from cell 1 to cell 2. So these extracellular messengers are having an action on a different cell. They're being released by cell 1. They're having an action on cell 2. When that's the case, these are called paracrines. And the way I sort of remember this is that para sounds like a pair of, so there are two cells involved. Paracrines. Paracrines are extracellular messengers that are released from one cell, have an effect on another cell. This cell that they have an effect on is in close proximity. Unlike hormones that have to travel a far distance in the blood, paracrines, when they're released from cell 1 to cell 2, cell 1 and cell 2 are close together. Unlike paracrines, there's another type of local mediator called an autocrine. And in this case, we have our extracellular messengers that get released from the cell, and they act on the same cell. So an autocrine, auto is self. In this case, this messenger comes and acts on the same cell. So the messenger is released from the cell. Oh, that doesn't have, messenger doesn't have an E on the end. The messenger is released from cell 1 and acts on cell 1. This is an autocrine. As you can probably imagine, if a cell is releasing something, releasing an extracellular messenger out into the environment to act on itself, it very often also acts on nearby cells. So these messengers, many messengers, extracellular messengers, are often both paracrine and autocrine. So if it gets released into the environment and this receptor is also on nearby cells, it's going to have an action on the nearby cells as well. So this autocrine messenger then becomes a paracrine messenger. So how do we classify them chemically? And then this hopefully sounds familiar. We have water-soluble extracellular messengers, and then we have lipid-soluble extracellular messengers. And this probably sounds a little bit familiar from when we were talking about the chemical composition of our bodies. Water-soluble and lipid-soluble. And if we think about what things can pass through the membrane, we're going to come back to this. So think about, if it's lipid-soluble, can it pass through the lipid bilayer? If it's water-soluble, can it pass through the lipid bilayer? Which of these things is going to be passing through the lipid bilayer? Hormones can travel in two different forms, and we'll talk about this a lot more in the endocrine system, but we're just going to preview this right now. They, depending on what type of hormone they are, they can travel freely in the blood, or they have to be wrapped up in a transport protein so that they can travel in the blood. So if these are the lipid soluble hormones, and we're going to come back and we're going to talk a lot more about that during the endocrine system. Receptors. All right, so we have messengers, which is what we've been talking about. But we've sort of hinted at the fact that a messenger isn't going to do any good unless it can talk to something. So the way we can think about this, or the way I think about it, is you have a key, and that's the messenger, and then you have a lock, and that's the receptor. You can have all the keys in the world. If you don't have the correct lock, you're not unlocking anything. You can have all the locks in the world. Without the correct key, you can't open that door. And that's what messengers and receptors are like. So we talked about our messengers, kind of just introduce them, and we're going to talk throughout the whole semester a great deal more, get into a lot more detail about these things that we've just touched on. But now let's talk about receptors. So receptors are kind of like, the way I think of them is like the lock, whereas the messengers are like the key. So a chemical messenger, in order to exert an effect, in order to do anything, there has to be a receptor on its target cell. So in order for that key to do anything, that chemical messenger to do anything, there has to be a receptor, the correct receptor on its target cell. So key in the lock analogy again. The receptors can be in two places. They can be on the cell membrane, or they can be inside the cell. And what determines whether they're on the cell membrane or inside the cell is what type of messenger. And I'm gonna have an image for this in a minute. Is the type of messenger. Is the messenger water soluble? Or is it lipid soluble? If it's water soluble, just kind of stop and think about it for a second. Is the messenger going to be on the membrane or inside the cell? The receptor, I apologize. If the messenger is water soluble, is the receptor going to be on the cell membrane, or is it going to be inside the cell? And then if the messenger is lipid soluble, same thing. Is it going to be on the cell membrane, or is it going to be inside the cell? Think on that. We're going to come back to that. Some important characteristics of receptors: they are specific. Meaning, if we release this messenger represented by these little blue balls, so all these little blue balls are being released from cell 1. They get released just everywhere. Into the environment, maybe into the intracellular fluid, or maybe they're getting released into the blood if it's a hormone. How do we prevent every cell from reacting to that messenger that's being released into the environment? Well, the way we prevent that, or the way we specify our reaction, is that receptors are very specific to their chemical messenger. So if this chemical messenger is released into the environment, it comes and it tries to bind over here to the receptor on the, apparently we're not animating correctly. Just kidding. Let's see if that'll work. Oh. Okay, my animations aren't working. So if this little guy here tries to come and bind to this receptor over on the left that's this green color, that receptor, it doesn't match with that messenger, and it says, nope, you're out of here, dude. Same thing over here. We try and come and bind, it doesn't match. It's not the right key for that lock, so it gets rejected. It doesn't bind to the receptor. We have to have the correct key and lock. We have to have the right messenger combined to the correct receptor in order for anything to happen. That's specificity. Then we have receptor location that I was hinting at earlier, that I said, let's think about that for a second. Based on the type of chemical messenger, that's going to determine where the receptor is located. And so our types are two broad chemical categories were water soluble and lipid soluble. And then our receptors can be in different locations. Our receptors can be on the cell membrane, or they can be inside the cell. And there are two locations inside the cell. They can be in the cytosol, or they can be inside the nucleus. So what we want to think about here is if we have a chemical messenger that's water soluble versus a chemical messenger that's lipid soluble, these are getting released. The lipid soluble chemical messengers are able to pass through the lipid bilayer, right? Lipid soluble, they can go in through the lipid bilayer, and then they can bind with their receptors that are either in the cytosol or in the nucleus. Whereas water soluble, those can't pass through that lipid bilayer. Remember, we saw that image in one of the earlier lectures where if you pour oil into water, it beads up, right? And remember that this bilayer is a lipid bilayer. So if we take water soluble things, so polar things, and we try to pass those through the lipid bilayer, that can't happen. They don't like each other. So our water soluble chemical messengers are going to have receptors on the cell membrane, so that water soluble chemical messenger doesn't have to pass through into the cell to deliver its message. Is kind of the way we can think about it. So lipid soluble is their receptors are going to be inside the cell. And water soluble are going to be on the membrane. For lipid soluble, there are occasionally exceptions. So just because it can pass into the cell doesn't mean it has to. So you do occasionally find, so they're sometimes on the membrane. But if a receptor is inside the cell, its chemical messenger cannot be water soluble. Cannot. Absolutely cannot be water soluble. I think I put, let's see. No, okay. And we'll talk more about that when we get to hormones. I thought maybe I put a little outline here, but I didn't. I decided to wait. Okay, receptor location. So what are the results of the interaction with membrane receptors versus intracellular receptors? Membrane-bound receptors. So here's our cell, and the receptor is on the surface of the cell, and its water-soluble chemical messenger comes and binds. What that does is that causes a reaction inside the cell, and this is a cascade. It's a cascade of events that we will talk about later in more detail. But the big kind of take-home message from this is that this cascade is fast. If you have a water-soluble receptor binding with a water-soluble messenger binding with a membrane-bound receptor, you're going to get a fast reaction. Why is that? Well, it's fast relative to the other option. The other option, remember, is that our receptor is inside the cell. And when our receptor is inside the cell, particularly when it's inside the nucleus, and our lipid-soluble messenger comes and binds, what that triggers is transcription and translation. So this is that central dogma. We'll come back to this again with hormones. The central dogma of genetics is that we go from DNA to RNA to protein. So we're transcribing into RNA, and then we translate into the protein, and remember, the protein is what actually does the work. So because all of this has to happen, because when we bind with a receptor in the cell, particularly in the nucleus, we have to go through transcription and then translation and make a protein, this is a slow response. Fast, when we have our water-soluble messengers binding a membrane-bound receptor, because this cascade, what it's doing is it's triggering an action that's already been pre-prepared, so those proteins have already been made. Whereas if we're in the nucleus, we're making new protein, and that's slow, and we're going to revisit that concept again. There are lots of different types of receptors, and again, we'll revisit this again. You've seen the channel receptors. We've talked about this one already, or a channel protein. So what can happen is that this channel protein that we discussed previously, it can have a chemical messenger come and bind, and I'm drawing this blue because these are in the membrane, so this is going to be a water soluble because these are not inside the cell. So its messenger comes and binds, and what that does is that opens this channel, and then that allows whatever the thing is that's supposed to come through that channel to pass through. Then we have this whole group over here on the right, and these are second messenger systems. And second messenger systems are considerably more complex than the channel receptors. In a second messenger system, you have a receptor come and bind. So the receptor comes and binds, and what that does is that sets off a cascade of events that requires more than just the protein that's embedded in the membrane. Over here with our channel, the only thing we needed to talk to make anything happen was that channel protein itself. With a second messenger system, in every case, you have to have a second messenger in addition to the protein that's embedded inside the membrane, and that's why these are called second messenger systems. Just introducing those, and we're going to come back to those in more detail. Okay, receptor agonists and antagonists. Stop and think for a second. What do those words make you think of? An agonist, or particularly an antagonist, is probably something that's pretty familiar. First, we're going to be talking about agonists. Here in our image, this is our receptor. And then this is our messenger, also called a ligand sometimes. And this is what's called our primary ligand. This is the thing that is you could think of it as supposed to trigger the receptor. And what happens when that ligand binds is it comes, it binds, and it triggers an intracellular response. So it tells the cell, do your thing, do the thing that you're supposed to do. That is the primary ligand. What an agonist is something that's not the primary ligand that causes the same effect, causes the same thing to happen. It triggers the intracellular response. That is an agonist. Whereas an antagonist does the opposite. So here's our primary ligand. It triggers a response. or a primary messenger is another way we could say that. An agonist, rather than triggering an intracellular response, it prevents an intracellular response. So when an agonist binds to that receptor, it fits the receptor, but nothing happens inside the cell. So what this does, this can be preventative. So it can stop reactions from happening. And almost every drug that we use is one of these two things. It's an agonist or it's an antagonist. It's mimicking the primary ligand, and it's either making something happen or stopping something from happening. So what I'd like you to think about now is the drug bromocriptine is used to treat pituitary tumors. It binds dopamine receptors and it alters enzyme activity in the cell. So is bromocriptine an agonist or an antagonist? And to which category of membrane receptor must it bind? So thinking about the different broad categories that we talked about with regard to membrane receptors.